All right, today we're going to tackle one of the most revolutionary tools in all of modern science, PCR. You've probably heard the term thrown around a lot, especially over the last few years. But what is it really? And how in the world does it actually work? Well, let's zoom in and find out. Okay, so picture this. You're a detective. You're standing in front of a library with millions of books, and you need to find one specific sentence hidden somewhere in just one of them. That's kind of the challenge scientists face with DNA. So how do you find that one tiny, microscopic clue in a vast, overwhelming sea of genetic code? Well, the scientist who invented PCR, Kary Mullis, had this incredibly simple but brilliant idea. He called it molecular phototyping. His thinking was, hey, instead of trying to find that one original sentence, why not just make billions of copies of it until it's pretty much the only thing you can see? Let's put that another way. A much better way to think about it is like a molecular photocopier. That's what PCR is. It takes a tiny, targeted piece of DNA, your single clue, and it just amplifies it, making millions or even billions of copies in just a couple of hours. It literally makes the invisible visible.
So to really get how powerful this is, we've got to start with the original blueprint. Let's break down how the first version, what we call conventional PCR, actually works.
Right, so here's the official textbook definition. It's a test tube system for DNA replication. Now, the two most important words there are selective and amplified. You're not just copying everything in sight, you're picking a very specific target and then making a ridiculous number of copies of just that little piece.
But here's the thing. To make this whole process run automatically, scientists needed a kind of magic ingredient. They needed an enzyme that could survive being boiled over and over and over again. And the answer, amazingly, wasn't in some high-tech lab. It was in the boiling hot springs of a geyser.
And this is the real hero of our story. A tiny microbe called Thermus aquaticus. This little guy loves living in those geysers, and it produces a special DNA copying enzyme, Taq polymerase, that doesn't get destroyed by extreme heat. Finding this was the absolute game changer that made automated PCR possible. So the whole process is just a three-step cycle, repeated again and again. First step, denaturation. We crank the heat up to about 95 degrees Celsius, and that separates the DNA into two single strands. Second, annealing. We cool things down, which lets these tiny little guide sequences, we call them primers, latch onto the exact spot we want to copy. And third, extension. We raise the temperature to about 72 degrees, and our hero, Taq polymerase, gets to work, building a brand new strand of DNA. And boom, that's one cycle done.
Now, get this, you don't just do that once. You repeat that cycle, say, 30 or 40 times. The number of copies doubles each time. 1 becomes 2, 2 becomes 4, 4 becomes 8. It's exponential. This growth is just unbelievably powerful. Within a couple of hours, you can go from a single molecule of DNA to over 1 billion copies.
But you know how science is, right? It never just stops. The minute conventional PCR was working, researchers started asking new questions. And that led to a whole family of upgraded PCR techniques, each one designed to solve a new, different kind of problem. So, the first big hurdle they ran into was that the original blueprint only works on DNA. But what if the genetic stuff you need to study is RNA? This is a huge deal, especially when you're working with a ton of different viruses, for example.
The solution was honestly beautiful in its simplicity. It's called RT-PCR. Scientists just added one step at the very beginning. They use an enzyme called reverse transcriptase to make a DNA copy of the RNA. Once you have that DNA copy, or cDNA, you just run a totally normal PCR on it to amplify it. And just like that, problem solved.
Okay, but that led to another question. Conventional PCR is great at telling you if your target is there, but it can't tell you how much of it you started with. You just get an answer at the very end. So, the next big question was, how can we literally watch the copies being made and count them as it's happening?
And this is where real-time PCR, or qPCR, comes into the picture. The big difference is right there in the name. Instead of waiting till the very end to see what happened, qPCR amplifies the DNA and counts it at the same time, using a fluorescent signal that gets brighter as more copies are made. You can see the results in real time.
So how does it actually do that? Well, it's pretty clever. The reaction mix has these special fluorescent probes. As our enzyme, Taq polymerase, copies the DNA, it bumps into these probes and breaks them, which releases a little flash of light. So the more DNA you make, the brighter the tube glows. A machine is just measuring that glow cycle by cycle, which lets you count the DNA with incredible precision.
All right, last big problem to solve: efficiency. Let's say you have one tiny, precious sample, and you need to look for 10 different things in it. Running 10 separate tests would take forever, cost a ton of money, and you'd probably use up your whole sample. So what do?
And the answer? It's called multiplex PCR. It's pretty much what it sounds like. You just throw multiple different sets of those guide primers into one single reaction tube. Each primer set is designed to find and copy a different target. Think about it. It's like running dozens of different tests all at the same time in one tiny tube. It's incredibly efficient.
So, we've gone through the science, we've seen how it evolved, but what does this all mean for us? In the real world, why does this molecular photocopier matter so much?
Honestly, the list of applications is just mind-blowing. In medicine, it's how we detect viruses and bacteria, how doctors monitor viral loads for diseases like HIV, how we screen donated blood to keep it safe. In forensics, it's the engine behind DNA fingerprinting. It's used in cancer therapy, in agriculture. Heck, it's even used to study DNA from ancient fossils and in dentistry. I mean, it is literally everywhere.
And the amazing thing is, the story is far from over. PCR isn't some old invention sitting on a shelf. It's a living, breathing platform for discovery that just keeps evolving.
I mean, just look at this journey. From that first idea back in 1983 to the game-changing upgrades in the 90s to today, where scientists are using over 37 different types of PCR. And each one is like a custom-built tool for a super specialized job. From analyzing the DNA in a single cell to something called suicide PCR, used to study ancient DNA without contamination. It's incredible.
And that really brings us to the big question, doesn't it? For more than 4 decades, this amazing technology has completely changed what we thought was possible in science. The real question isn't just about what PCR has already done, but what will this ever-evolving molecular photocopier make possible next?